Well, welcome everyone to this co-current session of Atlas, the Human Cell Atlas Asia. My name is Shin. I'm a team leader at Riken Yokohama, and I'll be co-chairing this session with Katy. So maybe Katy can give a brief introduction.
I'm Katy Borner, Indiana University, and it's a true pleasure to be here. I'm part of the HubMap team, which is the NIH-funded effort to create a reference atlas of the human body. Great.
So we have a very exciting lineup of speakers today, Katy, Longqi, and Xinhua, and we will be covering quite diverse topics, but we hope to have a discussion of how we can study and build atlases in an effective way that can have a good integration of different data sets in a meaningful way. Especially the human body is complex, and it's not just cells, but it's made out of tissues and organs. And how we can reconstruct this in a meaningful way is a big challenge for both Human Cell Atlas as well as HubMap and other consortiums. So hopefully through today's talks that we'll have good insights into how to initiate this effort. So I've asked each speaker to speak about 15 minutes or so, and we'll have about 10 minutes for Q&A. And hopefully we will finish the session at 11:50 Beijing time. So with further ado, let's get started with our first speaker. Let me give a quick broad introduction. Professor Katy Borner has a MS in electrical engineering from University of Technology in Leipzig and got a PhD in computer sciences from University of Kaiserslautern. She's a distinguished professor of engineering and information science in Department of Intelligent Systems Engineering and Information Science, School of Informatics, Computing, and Engineering at Indiana University. As you have heard, she's part of a HubMap project, and she is particularly interested in the formalization, measurement, and systematic improvement of people's data visualization literacy. So I think we're gonna hear a lot her efforts in the next 15 minutes or so. So Katy, please.
Okay, let me share my screen here. All right, you should all see the title slide. This is a presentation of a much larger team. It's really not just me, which is doing all this work. There's a lot of PhD students and staff members. And on the left-hand side, you see a picture of the first all-hands meeting of the HubMap team. In the meantime, we have grown to 300 participants. We just recently onboarded 11 new teams. And we work very closely with the tissue mapping centers and also 3D modeling experts to create an anatomically explicit reference atlas. Those of you which don't know much about HubMap, it's the ambition to create an open global framework for comprehensively mapping the human body at cellular resolution, so very similar to HCA. There are a number of links here that provide more information on the overall effort. We also just had our first data release, so you can see the HubMap data on the portal. I just posted the link to these slides into the chat log. So those of you which want to click on links and get the slides this way also are most welcome to download the slides and follow along this way. As for HubMap, we have a number of tissue mapping centers. We have transformative technology development centers and rapid technology implementation teams. And all of these different teams create tissue data sets. There are many different assay types. All this data is compiled and harmonized. And actually, Rahul Sahit, who is currently presenting in a parallel session, is leading the other mapping center, which is doing a lot of the data harmonization work and research. And then all of us are involved in map generation, and it's not just UMaps and TSNE plots, but it's also anatomically explicit tissue maps and 3D body maps. And then, of course, this data gets stored and disseminated so that anyone can access it. And again, we just onboarded 11 new teams, so that's quite a large set of experts. These are some of the organs we initially focus on. And again, we have many different types of assays from genomics to epigenomics to transcriptomics, proteomics, metabolomics. So many of us are trying to become experts as quickly as we can in all the new technologies and the different types and ways of identifying which single cells types are involved in what kind of tissue. We also have multiplex spatial assays, which is especially fascinating. And then ultimately, out of all these image stacks, there is the ambition to understand better anatomy, histology, and also, of course, cell populations, and to create a common coordinate framework. And again, Rahul is presenting right now in parallel, and he's the lead for the MCA New York Genomics Center mapping center. So there are 2 mapping centers within HubMap. If you would like to know more about HubMap, we have an entire MOOC, a massively open online course that you should share with your students and with yourself, and maybe also with your family members. We really want this to be widely disseminated. And some of the initial modules provide an overview. They provide an information on how the tissue data actually comes into existence. Rahul has an introduction to Surat. Our team has an introduction to CCF ontologies, 3D reference objects, and user interfaces. There's also information on how to use the current portal and how to open consent your data and why. And so this link gets you there. And I think also in the abstract for my talk, you will get the link. And feel free to repost the link to these slides if you would like to have them. So what is a CCF? For the purpose of HubMap, currently we define a CCF as a system that consists of ontologies and reference object libraries and computer software, but also training materials that do 3 things. They allow registration of new tissue, including user interfaces that support that. They also allow to map data from multiple participants, multiple labs, multiple technologies. And they make it possible to explore HubMap and other data at multiple levels, from the entire human body, which is quite large, down to the single cell level, which is quite small. So that is really a challenge also for the user interface design and data visualization design. And that's where our expertise in data visualization comes in. Initially, we have decided to have a nested approach. So you will have cells inside of so-called functional tissue units, inside of larger functional tissue units and anatomical structures inside of organs, and the organs are referenced and mapped into the human body. And as you see, different types of anatomical structures and cell types are relevant for different zoom levels, if you wish. Just like a map of the US wouldn't show you our neighborhood, which I have out here in the front yard, but it would only show the major roads. Similarly, here you get to see certain types of information only as you zoom in. In order to facilitate this, we have embarked on collecting so-called ASCT+B tables. So these are anatomical structures, cell types, and biomarker tables, which really aim to capture the autonomy that you just saw, cell types, but also major biomarkers. And it's non-trivial to arrive at nomenclature that everybody agrees upon. This is really a huge community effort. And the initial tables like this were actually compiled via the KPMP, the Kidney Precision Medicine effort. And these tables are quite large for entire organs. But we also, in the recent HCA NIH meeting in March this year, realized that there are many, many different consortia, which are here the rows, which care about many, many different organs, which are the rows here. So it's rows by columns. And then you also get to see what efforts care about what organs. And you can just sum them up. And so HCA definitely cares about the most organs right now. But then there are also other efforts which also care about quite a number of organs. And all of us are interested in these kind of correlations between anatomical structures and the cell types which are inside of those, and then biomarkers that are used to identify cell types. And then there's a big effort on agreeing on the cell type names and states. And so since March, we have been meeting on a monthly basis, also bringing in ontology experts, because ultimately these ontology terms have to link back to the existing ontologies, trying to understand which ontologies are most relevant, what can be used in these ontologies, and what new terms have to be added to these ontologies. And so we meet monthly, we review and approve existing tables. We formalize and unify the table design language, and I would love to see more experts from Asia joining these meetings. The next meetings are on November 5 and December 3rd from 1:30 to 2:30 EST. That might not be a perfect time for you, but you can also see all the recordings. So all the meetings have been recorded, and you can also bring in your presentations as recordings. You're welcome to register if you're an expert and would like to participate, and then we send you more information. The tables ultimately can be visualized. So here you have anatomical structures in red, cell types in blue, and biomarkers in green. And you can also see the ASCT+B across all the organs, and that very quickly helps identify differences in nomenclature and helps resolve those. Ultimately, these cell types and cell names have to match up with Rahul's efforts to provide reference atlases for different organs, and then make it possible for others to upload new data sets and to basically automatically transfer these labels over to new data sets, which basically creates a de facto standard for cell type naming. So his new Azimuth system, hopefully you get to check it out, is very powerful in actually helping us agree on cell type namings. And he is using the cell ontology as we do for the ASCT+B tables. Currently, we have these organs, and you also get to see here the number of anatomical structures, the number of cell types, number of biomarkers. And then, of course, we are very interested in the bimodal linkages between, for instance, anatomical structures and cell types and biomarkers. And so as time goes on, these numbers increase. Sometimes they decrease because we merge namings, but oftentimes they just grow. And we also now have a standard operating procedure for the construction of those tables. So if you don't see your favorite organ listed yet, there's hope. You can just work with us to get your table added. And we would very much welcome more contribution from Asia. These tables are then used to identify ontologies, not just the anatomical structures patonymies, but also cell type ontologies, and to arrive on 3D object libraries. And so for the ontologies, there's now also a paper on arXiv, which demonstrates how we interlink the spatial ontology to the semantic ontology to the clinical ontology, because it matters if you're male or female or a certain age, or if you have smoked or not, or if you're pregnant or not. It will matter in terms of what cell types will be found. We also use that patonymy then in the HubMap portal, which you can use to basically see what tissue blocks have been registered inside of what organs. And then you can click on those registered tissue blocks and you get to see what assay types have been run. And you can click on any of these thumbnails on the right-hand side and you go over to more information, but also to ultimately Vitesse by Niels Gelenborg's team, which helps you to just zoom into these tissue slices. And then on the reference object libraries and there are very interesting efforts also in Asia to make it easier to register tissue blocks that are currently typically captured via photos, such as what you see here. So this is a kidney and you cut out a larger part of it. And then you might send off one of these smaller pieces to one lab and the next one to another lab. And the next one is bulk, the next one is spatially explicit assay types. So these blocks all go to very different types of workflows, but then have to be recomposed so that we actually understand how different cell compositions exist across the different parts of the human body. For the first portal release, we registered 48 tissue blocks for kidney and for spleen, and you see the registration areas here and also the size of the tissue blocks. I know some of you might work with biopsy data. We can also register that, and it would look much smaller than the blocks you see here. This is the initial interface we used for the initial portal release. In the meantime, we have expanded the set of reference objects and also the level of detail that these reference organs have. But ultimately, it's the same process. You have cells inside of functional tissue units. The functional tissue units are part of these tissues blocks and section blocks. And then the tissue blocks are registered inside of reference organs, and the organs are registered inside of the human body, and everything is registered into the Atlas reference system for HubMap. And so if you have reference objects that you're working with, we would love to know about it and also add them to this reference body. We recently added the Allen brain, 3D human brain, to that reference body, because we believe that even if HubMap doesn't have the mandate to actually deal with brain data, it is just sad not to have a brain in that reference body. So we very much welcome collaborations on these reference organs. Here you get to see the new and revised registration user interface that will become available in 2 weeks. Here you will have support for colon, also for spleen, for kidney, and for heart. You have also a number of extraction sites that you can now use to register your tissue. And ultimately, if you register tissue inside of these reference organs, automatically ontology terms are generated. And these are ontology URLs that lead directly to Uberon, which you can also then add your own anatomical structure names to those if you don't have all of them listed yet. And then for HubMap, we have such registration user interface as part of the ingest portal, but we will also make it available for anyone else to use. And then you can download your own registration data as a JSON file and use it in your own portal, or you can come into the HubMap portal and make your data available there. And again, we also have a standard operating procedure for the creation of these reference organs. So if you would like to benefit from it, it's linked from here. Ultimately, then you see these reference blocks inside of the reference body, and we have a male and a female version of it. And if you click on it, you get to see the tissue blocks that exist and also the tissue sections. And then if you click in one of the tissue sections, you get to go over to Niels Gehlenborg's Vitesse viewer, which then lets you turn on and off certain types of expression patterns that are also in the data. Going forward, we are very interested to identify functional tissue units across different organs and focus on those and help understand if they differ, if they are from the upper part of the kidney, for instance, or the lower pole of the kidney, if they are from male or female, and how this differs as we age. And so again, if you're working on functional tissue units and their cell type compositions and sizes and shapes, I would love to hear from you. Ultimately, in the VH MOOCs, there's information on how you can register your own tissue blocks, but also on how to manually annotate human tissue. So please feel free to come into the Visible Human MOOC and learn with us. We also have an approach to use the vasculature as a coordinate system to map all the cells in the human body. And so for that particular line of work, we are very interested to connect with teams that focus on vasculature and cell types near vasculature, because ultimately we will need that to understand how we can use this vasculature as one of the systems, in addition to the nervous system and the lymph system, to really understand how different cells are distributed in our human bodies. So therefore, we also these ASCT+B tables for different system here for the vasculature. So just a checklist for everybody who would like to become part of this effort. There is an ASCT+B table effort and meetings, and you're very welcome to join those. If you would like to use the new registration user interface, there is a link here on how to do that. We have the exploration user interface that now is also showing data from other consortia, for instance, the Kidney Precision Medicine project that uses mostly biopsy data. And for functional tissue unit segmentation and the VCCF, as we call it, we would love to connect if this is of interest to you. And if you have questions on any of this, please feel free to email us under info@ccf@indiana.edu. We did have a set of questions, and if I have time, here are my first answers to those questions which Jay posed to us this morning. Do we want to do this now, or is this already going into the Q&A time, Jay?
Well, you feel free to raise them, yes.
Okay, so Jay this morning sent us an email to all the panelists and asked us to answer these questions. And I thought I would just put them on a slide. So one question is how to build an anatomical structure-based atlas. So how do you go about it? Because we are all so different. We might have different heights and sizes and ages and sexes and ethnicities. How do you go about having one reference atlas? How do you parameterize this? And then second is what strategy exists to define cell types and cell states in the context of building such an anatomical atlas. And I showed you what we do in HubMap, and as a much larger cross-consortium effort, we have these anatomical structures as pathonomies, we have these cell types, and then we try to understand what biomarkers and what technologies are best to ultimately agree on cell types and states. And then a question, are biomarkers sufficient, or should we define cells and cell states relative to 3D space and age and disease, so that we all can better understand cell types and their function and what happens when there is disease. And how do we best combine our efforts, given that we live in different time zones and we speak different language and potentially have different funding mechanisms. And so my first answers is that it would be wonderful to agree on names and just a set of anatomical structures, cell types, biomarkers for these 30+ organs that seem to be the focus of research and development right now. That we then would work very closely with the ontology experts to revise the existing ontologies as needed. We could all collaborate on these 3D scaffolds. We, for instance, collaborate very closely with the Spark team now, and Peter Hunter's team in New Zealand at the University of Auckland is very active there. In Japan, there is the body parts 3D effort, which is it's absolutely phenomenal what has been done there. And there might be other efforts that I don't know yet about, but I would love to connect, because it's a lot of work to just do one organ, and doing 30, I think you have to collaborate. No single team has the resources to do them all. And then when we have ontologies and we have 3D scaffolds, how do we best register new tissue blocks using, for instance, the registration user interface I just showed you? And then how to best explore this data in a meaningful way, not only by experts, but also in classroom settings, so that the next generation gets to benefit from all that effort. And so I showed you our exploration user interface, but there might be other approaches of how to better make that data available. And I would love to learn more about those. So okay, maybe I should stop sharing here and see if there's if there are questions.
Great, thank you very much, Katy. That was an excellent overview of just tremendous efforts of HubMap and really stitching together this whole body together in a very meaningful way. I'd be happy to raise any questions that are coming from the audience. So if you have not seen the chat, feel free to ask questions using the Q&A button that you can find at the bottom of the screen, and I'd be happy to read them out to Katy. So maybe in the process while the attendees are formulating questions, maybe I can ask a few questions in advance. So you have, most of these are currently based on single cell genomics and spatial transcriptomics data, I guess. You have in the earlier slides, you mentioned some proteomics and maybe metabolomics or others. So what is the direction of HubMap in integrating proteomics and beyond just RNA expression level?
So I believe I can send a link to everybody. So if you go to that website, you will go to the HubMap portal, and you will see all the many different types of assays that we are currently offering data from for the HubMap effort. As you might have seen, Rahul's team has used the site-seek data to create these reference atlases. And ultimately, you can also use RNA-seq data to now map onto those reference maps, which his team is creating. You might also have seen that we have quite a number of lipidomics data. And going back here in my slides, I wonder if I should bring them up again, but there is genomics data, epigenomics data, transcriptomics data, proteomics data, and metabolomics data that we have. And we are not harmonizing and using all of this for the CCF effort yet. We work currently very closely with the kidney team and the colon team, which is the Vanderbilt University team and the University of Stanford team. But as we get more data to come in, and the next data release will be there for Christmas, so there's a Christmas data release, and then it's a 6-month cycle. As these data sets become available, we will use them for CCF design, but we are still in the process of also learning what kind of data is useful for what types of cell type identification.
Great. Thank you. Maybe I'll ask one quick question. So you mentioned anatomical structure and cell type and biomarker. So these are in that particular order, I guess. So these biomarkers define a cell type. Do you try to define a biomarker that defines anatomical structure? So are there
indirectly?
Indirectly, yeah.
Yeah, actually in the ASCT+B reporter, which is another link that I can share here. So the beauty is that you can actually try out all these different interfaces. So in the reporter interface, if you go to visualization, you will see that you can actually hover over a cell type, and you will get to see all the biomarkers that are connected to it. You will also get to see in which anatomical structures that cell type lives. So if you have an endothelial cell, that might be part of multiple anatomical structures. You might also go to the all organs view, in which case you get to see that CD31 is used across many different organ types to identify endothelial cells. So you get to see across the organs what biomarkers are used, what cells are in existence across different anatomical structures. And hopefully that also leads to a more unifying nomenclature for cell types, but also for biomarkers.
Yeah. Excellent. I mean, it'd be great one day to use biomarkers to identify the location of the cells. Right? That'd be a nice.
And that should be possible. So the beauty is that with the HubMap portal, we will use these ASCT+B tables for search. So you will be very soon, not yet, but very soon you will be able to run spatial queries. So you can, for instance, pick one cell and get everything in a certain vicinity of the cell, or one functional tissue unit or one anatomical structure. And you will get to see what's in the vicinity, but then not only in the anatomical context, but also as part of the UMAP and TSNE plots that Rahul's team is producing. So you have both views, you have actually all three views. You have the ontology, you have the anatomical view, and you have the cell semantic space view, as I would call it, using UMAP or TSNE in your world typically. But there are many other ways to visualize his data sets, especially in a way that you could zoom in. And Rahul actually uses the cell ontology, which has a hierarchical structure. So you could come from very general cell types to very specific cell types. And as we learn more about different cell states and types, there might be multiple levels for potentially zooming in. And then in addition to all of this, you have the clinical data where you can request to see only data from Asian gentlemen or Asian ladies. And that would further subset and filter what you see in those three displays. And that's not yet in production, but we are working towards that kind of interface.
That sounds very exciting. So thank you, Katie, for your excellent presentation and response to questions.